Hi, my name is Katie Burch. I am a current biomedical sciences PhD student. I will be presenting my poster over exploratory analysis of estrogen-mediated gene expression and central leptin signaling pathways. To begin, we all know that obesity is a major problem for Americans, but particularly concerning is the greater incidence of obesity in postmenopausal women. So to address this concern, we used a microarray to look at the changes in gene expression from estrogen, within the leptin signaling pathway. Leptin is produced in your adipose or your fat tissue, and therefore, with an increase in body weight, you have an associated increase in circulating leptin. So to summarize my methods, I had 6 rats, and they were all ovariectomized or had their ovaries removed, so none of them were producing estrogen. And then half of them were treated with estrogen injections, while the other half was treated with oil injections as a control group. And you can see in this graph here that all rats' body weight goes up until the treated rats start receiving their injections, and their body weight starts going down, while the oil-treated rats' body weights keep going up. And with that increase in body weight, they have an increase in adipose tissue, which, as I said, releases leptin, and then that leptin then goes into the brain, particularly the hypothalamus, and the arcuate nucleus is where those leptin receptors are first or mainly received, which they contain these 2 major relevant neurons that express POMC-CART or MPY-RGRP. For the sake of time, I'm going to be focusing on the satiety center or the paraventricular nucleus, PVN. And you can see that the MPY expressing neuron is an orexigenic neuron. It tells you that you're hungry, you need to keep eating, and so therefore it sends a inhibitory signal to the PVN, which is denoted by the red arrow, and is received by the MPY1R. And then the POMC-CART neuron, which is an orexigenic neuron, tells you that, hey, you have enough energy stored in your body, so you should stop eating. So that is sends excitatory signals to the PVN, which is denoted by the green arrows. So when these signals are received in the PVN, then these neurotransmitters inside here, the BDNF or the CRH, they receive that signal and then they act accordingly. So I have all of these numbers. You can follow the pathway 1, 2, 3, 4, 5, etc. on this graph here, which shows the fold changes or the difference in the gene expressions between the estrogen treated and oil treated rats. And so if there is a positive number, that means it is upregulated in the estrogen-treated rats. They are expressing it more. So the more significant notes are the changes within the PVN, like I said, starting with the MPY1R receptor, which you can see by the graph down here, has a upregulation in estrogen-treated rats. And then the BDNF inside is the only one that I have a graph of that is upregulated in the oil. And so these are all anorexigenic neurohormones except for the MPY1R. So in conclusion, we did in fact see that the estrogen has an effect on these genes within these areas that we looked at, and therefore it is confirming that estrogen has a significant role in the leptin signaling pathway. But surprisingly, the MPY1R, which inhibits the satiety center, saying, hey, you need to keep eating, was also upregulated in the estrogen-treated rats, while the CRH, TRH, and oxytocin was also upregulated, saying you do have enough food. And the oil-treated rats which with their greater amount of adipose, do indeed attempt to activate the satiety center by upregulating the anorexigenic neurohormone of BDNF. future studies will look at more intracellular signaling within the PBN and look at other further areas such as the LH or the NTS, which are representing the downstream effects of these areas. Thank you.